Good evening. This is Smart Future AI Applications and Empowerment Series Forum. And this forum is initiated by 8 professional organizations in DMV area. And today we will continue our series 1, which is AI in biopharmaceutical industry. And today we are very honored to invite Dr. Zhirong Xie to give the talk. And his talk title is AI in Drug Development, from the technology to applications. So this is a very interesting, it should be the very interesting talk.
So I say good morning for people in Asia and good evening for people in US and Canada. And I also know that our audience have a very different background. Some people probably this AI expert and uh, want to learn the applications, AI applications in drug discovery and development. And some people that have very good experience in drug discovery and wondering how they can use AI in their work. So in today's talk, I will not present specific applications, with detail. I just give a high-level introduction on both the AI technologies and also drug development. And my goal is to help people from two sides to, better understand the another side, for the AI expert better understand the drug development, for the people from pharma and biotech better understand the technology. And I hope this talk can help the communications between two sides and also improve the collaboration between two sides. So in this talk, I will first give an introduction about the trend of AI technologies, and then discuss the application of the AI method in the drug discovery and development. And after that, I'll give a brief introduction about the public available data can be used to build the AI models. And then in the end, I also give some introduction about the organ on chip in drug discovery, which our company put effort on. So, when we talk about the AI, especially this round of AI, most time we are talking about the deep learning. Originally, the different deep neural network was designed for different type of tasks. For example, the convolutional neural network was used for the computer vision because the convolution can handle the 2D or 3D spatial information of the pixels. And the recurrent neural network is used to handle the sequential data, like the natural language processing. Also have the graph neural network uh, used to handle the graph data, such as we know like the social network or knowledge graph can be handled by graph network. And later on, Google group they proposed the architecture called transformer. Originally, this was used for natural language processing and did a very good job. And quickly, this technology was adopted into the computer vision field. We have the vision transformer. And now also, this was adopted in the graph neural network. We have a graph transformer. Another architecture was proposed recently, recent years, a few years ago, called Mamba, and as a alternation of the transformer, but still the transformer right now is still dominating this field. And for the AI models, we have two. Probably you can see we have two different models. One is the predictive model, and another one we call the generative model. The predictive AI forecasts the outcome based on the current, available data pattern, and this includes the method like prediction or classification. And it's easier to understand the prediction, but classification, when we classify, like different items or objects into different categories, we also see this prediction. And detection, like identify something in the image or in the text information or in the signal. It's also we call this a predict. We also use a predict model and segmentation, like a segment image segmentation or like a term or a sentence segmentation, and also treat as predict AI. And generative AI will create the new content based on the user request. For example, right now you can use a large language model and to create the images, new images or videos or audios, text, and can write software codes. So this all belong to the generative AI. So for the generative model, basically we can probably consider this like four different kind of model. And one we call one is the generative adversarial network GAN. And in this method, we train the two neural networks. One is called the generator, one is called the discriminator. And generator try to generate the pictures. For example, if in the computer vision field, generator try to generate the pictures which looks like real. And discriminators try to tell if this picture is real or fake. And these two networks fight each other, because the generator try to generate pictures to fool the discriminator, that the discriminator cannot tell it's real or fake. And discriminator try to, differentiate the generated pictures from the real images. So
Wow, how did that?
after they fight each other, and then in the end, the generator can generate the pictures looks very real. Another one is called variational autoencoder. The idea is if you have high dimensional data, and then you can use a neural network to layer by layer to get the latent space. And traditional autoencoders, after we directly from latent space reconstruct the data layer by layer and reconstruct original data, and such that the reconstructed data is the same or similar as the original data. But with our variational autoencoder, we not directly can reconstruct the data from the latent space. Instead, we just sample data from latent space based on certain statistical distributions, and based on these distributions, reconstruct data. In this case, it's reconstruct some new data. And also regressive model, mostly used in the natural language processing part. Like given some word in a sentence, and then it predicts the next word, and this can be used for like writing email, writing articles, and diffusion model. With the image, we can add noise to the add noise step by step, and then we can convert this image into noise, and then we train the neural network to reconstruct the image from the noise. So in this case, you give a noise, you can create the picture, and also this can be combined with the text information. You give text information, and then you can create the picture based on your request, text request. So over the last 10 years, the AI technology make a big or huge progress, from the task-specific models to the foundation models, and now we have a large language model and agentic AI. So originally, the AI was designed for the specific task. For example, in the computer vision field, we have different tasks such as image classification, where we classify the pictures into different categories, like the cat or dog. And in the object detection, we try to identify the object in the picture and then classify the object into different categories. And for structure segmentation, we try to classify the pixels of the image into different categories. For different tasks, we need to prepare different training data. For example, in the image classification, we need to prepare the image level annotation. We indicate like which category this picture belongs to. For object detection, we need to prepare the object level annotation. We need to give the location of the object, like the bounding box or mask of the object. and also give the category of the object. For structure segmentation, we need to give the annotation at the pixel level. We need to indicate every pixel, we need to indicate which structure this pixel belong to. And also for different tasks, we need to design different network architecture because their output are different. And then with this network architecture, we can train the model with the training data to optimize the parameters of this network. Normally, if we train the model for one specific task, and this model not works if you apply this to other task. For example, if you train the model for image classification, and probably not work directly for the structure segmentation. And even for structure segmentation, if you train the model to segment the liver on the CT image, probably will not work if you want to use this to segment the liver on the MRI image. So it's very task specific. And same thing for natural language processing. We also develop the specific models for specific tasks, like the name entity recognition. Here, we try to identify different terms in the sentence. Also, classify the terms into different category. And prediction, you predict the relationship between these different terms. And sentiment classification, you classify the sentence into like a positive, negative, neutral sentiment. And language translation, you can translate English into Chinese, Chinese into French, like something like this. And also, normally the model only works for the specific task. So since we have the transformer, and it's very easy to scale up the models. And previously, normally we had a model with like a million or 10 million parameters. But with the transformer, we have like billion or 10 billion parameters. But with large model, we always need a large amount of training data. And problem is most time we have a lot of data, but we don't have annotation. So the self-supervised learning was proposed for this purpose. And probably the next work is about in the natural language processing field is the BERT model. And in this model, they trained a very large transformer, the neural networks with a lot of transformers, and for the natural language processing. The idea is in a sentence, we have a lot of text information. In a sentence, they just mask out some word in the sentence. Like here, like how are, and then this word was masked, and doing today. So and then they train the model to predict what is the masked word. And then, after this, after train the model can predict this word. But the goal of training this model is not to predict the missing word. The goal is to learn the representations about the word or sentence or paragraph. So with this representation, after I train this model with this type of representation, we can use this in the downstream task and it works very well. And because this one, we do not need to use labeled data because we can find so many, textbooks, and, on the internet, so many text informations. You just mask some word out, randomly pick up some word, mask out, and train this, and then, you can get the model. So we call this a self-supervised learning. It's possible to train the model with a very large amount of data. And in image field, also the different model was proposed to learn the image representations. Here is just one example, the Dino model. And in this model, from one picture, and they can just crop this picture, sub-region of the pictures. And they can do the rotation or change the color or scale it. And then they train the two neural networks and identical neural networks. And so from the two view of this, the goal is from two view of the same picture, and the embedding of the feature learned from the two view are similar. So in this case, also we can use unlabeled pictures to train the neural network and to learn the representation of the pictures, and then this can be used on the downstream task. And besides the unsupervised, a self-supervised learning, also people use use the large amount of available data and to train some for with the supervised learning. This for this work they may train the model with a different type of data where there's some annotation available. Like this example, the uh, the model, AI model, was trained on different type of images like MRI and CT, X-ray, and digital pathology and the pictures, and they train the model to do the segmentation. And after the training, this model can work on many different type of images and do the segmentation, many different kind of organ or tissue segmentations. And also this work is very, this type of work, the foundation model is very useful to us because previously, if we want to develop the AI solution, always we need to prepare a large amount of data and make an annotation and then train the model only solve the specific problem. And in the medical field, the annotation is very expensive because you need the medical expertise to do the work. And with the foundation model, it's possible to directly use the model for our task. Like this, segmentation model, and many times probably can directly use to solve your problem. And also, it's possible, if it's work not very well, it's possible to just prepare a small amount of and label the data and fine-tune the model for the specific task. So this lower the bar to develop the AI solution.
Dr. Xie, there is a question in the chat.
Yeah.
Is Vision Transformer also a foundation model with self-supervised learning like BERT and Dino?
Oh, that's a very good question. Actually, it's depend on how you understand the vision transformer. If you simply refer to vision transformer for the model like a Dino model, pre-trained on the data, and then you can see this foundation model. But otherwise, the vision transformer we can reference just as the architecture of the network. So the architecture, we have parameters, but if it's not a trend, that you don't know the weight, the weight of network or the parameter of network, and then we not see this foundation model because we on the architecture, like the same thing as like if you have the formula without knowing like you have like for example AX plus B. If you don't know A and B, and you cannot use this to do anything. So that for that case, we will not say the vision transformer is a foundation model. I'm not sure if I answer your question. Okay, yeah. So now in the biomedical field, there's a lot of foundation model available. I mentioned the BERT model, and after this, people fine-tune this model on the biomedical document and get the BioBERT. And then later on, people apply the similar idea into other data, like the DNA sequence of single cell gene expressions and like a protein sequence. They mask out some piece of information and predict this information, and then they can use a model to learn the representations of these different like biological things. Like we have the DNA BERT, gene BERT, SC BERT, SC GPT, and protein BERT, anti-BERT. Anti-BERT is specifically for the antibody. And for the imaging field, we have the foundation models for cell images, like cell SAM, cell VTA. Also, we have the foundation models for digital pathology, like C-Transpath and H-Optimus, HIPT, PCON. Actually, there's a lot of models available. I didn't list all of them here because that's not the purpose. And for medical field, they also have like a Medison, Redimer, and MediImagePath. Yeah, actually, another thing I forgot to mention when we talk about the foundation model, like trained with supervised learning. One is the find that all the labels for the different type of image is one case. Another case is people right now they also find like the images with like a with the notes from like a radiology with a radiologist or with a pathologist, or maybe the pictures in the publication with the captions, and then they train this right now the technology allow us to align the text information with the images. So they treat the text information as a label of the image and then can train the model, with the text information as a label. So now, after foundation model, now we have the agentic AI. And, in the beginning, we have, as mentioned, we have large language models. And then we start use the like retrieval augmented generation and to address some issue in large language model. And now we have the agentic AI. So, actually large language models, since the first release of the ChatGPT a few years ago, now it's coming into our life, personal life. I think many people start use the large language model for question answering and help them to reading stuff and give summary or help them to write like an email, write other things, use the large language model. But we know the large language model also suffer a lot of issues like hallucinations. Many times they give us some wrong answers and also bias, because it's probably the trend on certain type of data. And then, if you give, in some scenario, it doesn't work. And also have a limited long-term memory. And, I think it's, like, direct user trained model it cannot remember everything correctly, and limited reasoning skills and limited knowledge. So to address some issue of these issues, people use retrieval augmented generation. The idea is when you ask a large language model to answer your question, you can also ask a large language model answer your questions based on some specific, the data, either the documents or other type of data, and give you the question. So in this way, we can help them to use the correct data to answer the question, and also make this question answering like more traceable and verifiable, and can be easier to explain, and also can improve the knowledge integration and reasoning. And now have the agentic AI and probably can work better than the RAG method. The agentic AI, we use large language model as like the brain to interact with different tools and different data to answer our question. And normally we can use this to recall to call the sub agent to interact with the tools for we first divided the larger task into small ones, subtask, and then call the different sub agent, interact with the tools to get the answer, and then later on put all this answer together step by step and do the reasoning and give us the answer. So this is just one example of the biomedical AI agent and developed by the Stanford team. And in this AI agent, they incorporated more than 100 software, 150 biological specialized tools, and 59 database. Here list some tools and database. You can see this BindingDB, the database, and the DESeq for the gene expression, differential gene expression analysis. There's a bioinformatics tool, Planck for the genetic analysis, and also you can see the scale image. This actually the Python package for the image analysis, BioPython, and admit prediction. A lot of tools available here. So when you ask a question to this agent will divide the question into subtasks, and then they call these tools to access the different database, or maybe search the data from the internet or from database, and then they call the specific tools to do the analysis on this data and then return the answers. After get the answers, then they can put these answers together, do the reasoning step by step, and give you the final answer. So for the chemistry, also we have the AI agent. Different group also developed AI agent for similar purpose. They call different tools. With a question, they can call different tools, do different tasks, and then return your answers, and then you can put them together, do the reasoning, get the final answers. And probably many people will think about to build the AI agent is very complex. Unfortunately, it's not the case. And right now, many organizations and groups that develop the tools to build this AI agent. And some tools is low code tools, and we can make it much easier to write code to configure the AI agent. Very easy to write some code to access, manage the data accessing or reasoning, different things and to do this. And some of like the launching and semantic kernel and different tools for this purpose. I also have some even no code tools. You do not need to even write code and to configure the AI agent like DeFi and other tools and to do this work and make our life much easier. So now it comes to the drug discovery and development. So in the here just this some a process of drug discovery and development. First, we need to do the target identification and validation. For this work, we need to understand the disease target biology, and we need to deal with different type of data like genetics data, genomics data, proteins, transcriptomics, epigenetics. And different data give us different information, so like how the gene affect the disease or how the variation of the sequence, the DNA sequence and affect the disease, and proteins interact with the disease, and also the genes, how what's the gene expression difference between the normal and abnormal tissues, and epigenetics, how the like even like non-coding sequence affect the gene expressions. And also need to understand the cellular signaling pathway and understand what caused the disease, and so we can find the target and use the target to treat based on target to treat the disease. Definitely for the identified target, also we need to study the druggability of the target. And after we have the target, then we can design the studies to screen large amount of compounds or ligand against the target. The first job is to build the assays, such as like protein or enzyme or cells. Then we screen large amount of compound against this like assay to find out like binding for the protein, find out the binding affinity, or find the enzyme activities or cell viabilities to see like how the compound affects the cell status, like can kill, if it can kill the compound, kill the cancer cell or suppress the cancer cell. And after finding the compound, then we need to optimize the structure of the compound to improve the potency of the compound's binding affinity with the target or specificity, like we want the compound only bind to the target for the disease, not bind to other target. And we also want compound meet some drug-like properties and have good as ADMET. That ADMET is absorption, distribution, metabolism, and excretion, and toxicity of that describes the how the compound behavior in the bodies, and then how if there's toxic issues, and this can be done using in vivo or animal studies. But in the lead optimization phase, most time people use the in vitro studies and for this purpose. And then preclinical study, we use animal to test the drug efficacy, toxicities, and selectivity and exposure. If the drug get into the disease area and binding to the target. Also the PK and the PD, and like how the body react to the drug, and how the drug what the drug did for does for the body. And we also need to choose the dose, prepare this for the clinical studies, and also develop the translational biomarker. That means we want to find a biomarker which can measure the treatment effect, and then this measurement can be used in clinical phase. And clinical phase actually is not only one phase, at least three-phase study. And phase 1 study, we study the drug safety, and also to see if the drug can cause any issue, and find the dose ranges for safety with safety concern. I also can study the PK and the PD of the drug. And phase 2, we evaluate efficacy and safety based on small amount of patient, as real patient, to see if the drug works as expected. Normally, we can call this a POC, as proof of concept, or POM, proof of mechanism. And after this phase 2 study, if you demonstrate good effect, and then we move to the phase 3 study to evaluate drug efficacy and safety on a large amount of patient. And after pass that, and we can seek the regulatory agency to get approval to market the drug. So, when we talk about the AI drug discovery, interestingly, most time people are talking about the early stage of AI drug discovery, like the target validation, target identification and validation, and drug design and optimization, and the very early stage. And this is very important, in the whole drug discovery and development. But also we need to be aware, and most of cost in drug discovery and development was spent on the clinical trial. Normally, this drug discovery and development is a long, very expensive long-term work. This by average, it takes more than 10 years and costs more than $1 billion. So, but most of the cost, more than 60% of the cost, including the time or the money, was spent on the clinical trials. And another issue is the clinical trial, the success rate is not very high. And normally it's between like 10 to 20% of the success rate. So to save the cost, we need to increase the success rate of the clinical trial. And if drug doesn't work, want to fail early, fail fast, terminate the program before it move to the clinical trial. And if the drug gets into the clinical trial, also we want to reduce, if we can use AI to reduce the time or sample size of the clinical trial, this can also save us a lot of money. So to do this, we need to get the right target to work on. And if the drug target is wrong, you will not get a good result. And then also we need to predict the molecular activity correctly before we move to the preclinical study or clinical study. And also we want to translate the in vitro or preclinical findings into the clinical outcome. Many times we get a positive results on the in vitro study and preclinical study, and then fail in the clinical study. And so if we can translate the result into clinical, it's also very helpful. Identify the right biomarker and can help us to identify identify right patient, and also measure the treatment effect. And definitely we need to select the right patient. This is can use the biomarker to select patient. Also use right now many people do the research using the natural language processing to help the recruitment of the patient, recruit the right patient in the clinical trial. And also need to choose the right dose. And also, another one is we need to measure the treatment effect accurately. Many times the clinical trial failed because there's not good biomarker or good measurement for the treatment effect. So AI can help us on all these type of works, at different stage of the drug discovery and development, and different type of data we collected, include the structure tables like the test result, or maybe like a count of gene expression or proteins, and many other structure tables. And also have the sequential data like the DNA, RNA, protein sequence. We have signals from EEG, ECG sensors like wearable device and other medical device. More importantly, we have a lot of text information like clinical notes. We have publications, we have regulatory documents, many text information there. And for imaging, we have the cellular images and for cell, to observe the cell response to different treatment or to the disease, and also digital pathology have information from the tissue level. Cellular image give us information at the cell level. Digital pathology give us information at the tissue level about the anatomy of the tissue with the disease or without the disease, and also the distribution of different types of cells in the tissues and the interaction between them. Medical imaging give us information about the organ. We have different types of medical imaging. We have MRI, PET, CT, ultrasound, X-ray, many different types of images. All of them have the strengths of and weakness for different purpose. Even MRI, we have different type of MRIs for different things. This can give us information about the anatomy of the organ or give us information about function of the organ. And also we have graph data like knowledge graph, which give us information about the relationship between drug, disease, target, pathway, and anatomy and the phenotype, this kind of informations. And also have the molecular structure, which can be represented as a graph. We can use the nodes, graph nodes to represent the atom. We can use the edge to represent the bond between the atoms. And then we can use the technology graph analysis to handle this data. So with all this data, we can use the different AI technologies to handle the data. Like computer vision can be used to handle the imaging data, and natural language processing not only used to handle the text data, can also be used to handle some sequential data. And graph network can be used graph data. And with these technologies and the data, we can get better diagnosis and better quantification. They quantify the data and to monitor disease progression, to monitor treatment effect, and also get a better prediction. And with this, we can better understand the disease and better understand the treatment, and also better understand the patient. So, the predictive model, we mentioned about the predictive model and the generative model. Predictive model can work in many places in drug discovery and development. From the target identification, this can be used to predict which gene or proteins or other things, how this cause the disease, and also can be used to validate the target. And then the compound screening, we may use the virtual screening before we do the high throughput experiment. And first, we predict which compound may bind to the target. And after that, we can select the compound and then to do the in vitro study to test how this compound works with the target. And for the compound optimization, also we need to use the model to predict the properties, activities of the drug. Normally, when we do the predictions, we always need some data collected in the later stage. For example, data collected in vitro study or collected in preclinical study or collected in clinical study to predict the compound properties and activities. And also from the in vitro study, also use a predictive model to measure the result. We collect maybe in vitro study, we may collect the image data, we may collect the bioassay or gene expressions, the different type of data. And we may use predictive model to do the better quantify this measurement, and also want to use measurement to predict, if we observe something in vitro, can we predict the preclinical result and the clinical outcome? And also in the preclinical studies, we collect many different type of data, and then we can use AI technologies to do the analysis. And definitely in clinical studies, we have a huge amount of data, and we can use AI technology to do analysis. And actually, I need to mention here, we mentioned AI actually is, the traditional method, like the statistical analysis and machine learning still play a very important role on this. So the AI just provide us additional powerful tool for the task. And generative model, I can also find a lot of applications in the field, like when we, for the drug design, definitely we need the generative models, drug design optimization. And also we can use it for the question answering. And not only for the question answering to the scientists, also maybe use this for patient, to engage the patient in clinical trial, make them to follow the protocol of the studies. And also can be used in document generation and QCs, and maybe use experimental study design. So to build the AI models, a lot of data, right now there's a lot of public data available. For example, for the molecular information, we have like PubChem, Campbell, and also protein, we have a PDB, Protein Data Bank. They have very rich data information. So for example, PubChem have more than 100 million compound, and also this with some bioassay test result and bioactivities, different kind of information. And also we have the data for the gene genomes and pathways, and like the DNA, mRNA, and copy number variations, protein expression on different disease and also on normal people, and pathway informations, and also have some clinical data. This data, Cancer Genome Atlas, the TCGA, not only have the genomics, omics data, they also have the lot of clinical information. Also have data for the drug targeting interactions, like open target, binding DB, drug target reactants. Have toxicity, the data for the toxicity information, like TOX21 and CTT and ECHA, also maintain the database, for the many chemical compound, and give information about different type of toxicity, like the acute type toxicity and corrosion and sensitization and genetic toxicities. A knowledge graph is very important data, and with the knowledge graph, the knowledge graph give us information about like a relationship between the drug target, disease pathway, anatomy, and this kind of information. And this is very important for us to understand, how the drug works, and also for the drug repurposing. And also, there are a lot of clinical studies, and this is not for the refer to clinical trial. Definitely, the clinical trial data is very, very important, but most times the clinical trial data is not available at patient level. But fortunately, we also have a lot of data, like natural history studies. We have the data at the individual, from individual patient on people, and like the UK Biobank, they have collected data. It's already almost 20 years. I think it's probably 18 or 17 years. And they collect data from hundreds of thousands of participants. And they have imaging data, genetics, and health linkage, and also have the biomarkers, activity monitor with a wearable device, and many other data. And this IDA is a database maintained by the Lonely Lab in I think the University of South California, and they also collect a lot of the imaging data. Most of them neuroscience studies, like this one, the ADNI study. They monitor thousands of normal people and patient, elderly people for more than 20 years with different kind of imaging data and other data available. And real-world data is also very helpful in drug discovery and development. Real-world data include the electric health record, EHR data, and claim data, claim and billing data from the insurance company. Also have like product disease registries, as some organizations build a registry as patient to contribute the data to study specific disease, and have patient-generated data, and like the data generated from the wearable device. And with this, this data, we can use this to optimize the clinical trial design. We can create the synthetic controls. Like in clinical trial, normally we need to have different arm, like with either placebo or treatment, or maybe compare a new drug with standard care. And most time we may use some real-world data to enrich the control group. And also we can evaluate the treatment efficacy in the real-world scenario, and identify the new indication. Many times from the real-world data, we can find that this drug was designed developed to treat one disease, but in the real-world situation and the data, we find that it can also treat another type of disease. And we always use the real-world data to monitor the drug safeties. And also we can use the real-world data to develop new biomarkers, either for the predictive biomarkers or prognostic biomarkers, or biomarkers to evaluate the safety. So although a lot of data available, but we know that data is not collected for the AI purpose. It's not only this public available data, even the data, historical data collected by the pharmaceutical company, the big pharma, the data original was not collected for AI purpose. So they always suffer some issues like quality, availability, standardization, space, imbalance, sparsity, silos. And that also, many times if you want to use the data, also have privacy issues, shareabilities. Here I just talk about like sparsity, historically when in the pharmaceutical company, they collect data just to evaluate for specific purpose, to evaluate like the efficacy, safety on certain situations. So most time, like we may test this drug, this type of drug on this type of bioassay, and test the other drug on the other bioassay. But most time we did not test all the drug on same bioassay. So in this case, we have lots of missing data. This makes it's very difficult to train the AI models. To solve this problem, right now, many organizations start to collect the data systematically. For example, they use the high throughput experiment and to collect to test the drug. Like you can put with the plate, like this plate can have like more than 1,000 wells in the plate. Each well you can put cells in the well, and then you can test different drugs like small molecular or big molecular or gene editing, put it into these wells, mix with the cells, and then to see how the cells responds to the treatment. And after that, you can collect like image, high content images with different like fluorescent stain. You can also collect the gene expression. You can also collect the bioassay from the wells. And one effort is, from the a broad institute that collects a large amount of they organize this activity and collect a large amount of data. And here, I didn't list all of the project, just some of them. You can see many studies that collect they test many drugs, like the the first one tested more than 300 drugs and 160 gene editings and collect a large amount of on certain cells like A549 and U2OS and collect 12 terabytes data and with 6 terabytes from imagings. And you can see another one, the bigger one is the CPG-003 tested more than 2,800 genes and compounds on this. And the one have the 16 CPG-16 and also collect more than 300 terabytes data. And so that we have they collect a lot of data with the same experiment and makes it easier to train the AI model. But the problem is, these cells are always cultured on the 3D on the traditional plates, the 2D, we call it the 2D model. And in this case, the cells cultured in the dish, they're not in the same environment as cells in the tissue, so they function differently as the in vivo. And also another one is for animal studies. Right now, historically, also have a lot of data collected from animal studies. But animal studies, we know that it's first, we cannot get a large amount of data. Secondly, the many diseases we don't have any model. Thirdly, the animals don't have same gene as human. Many times, we have positive result on the animal model, but failed in the clinical trial. So organ chip may be used to address some of these issues. The organ chip actually is just a multi-channel 3D device, which we can culture the cells, mimic the cell environment as the in vivo. So with the organ chip, we can create the tissue-tissue interface. We can do the microfluid control. We can co-culture different cells, like we can integrate the immune cells together with other cells, with cancer cells, and to model the disease. And with the disease model, we can test the drug and also maybe can use this for the personalized medicine. So this is just some Zeller product for high-throughput screening or to test the drug or drug efficacy safety. We have a 2-channel organ chip, and also we have 3-channel organ chip with different design. And like 2-channel organ chip, we have one like OCPlex 96. The one plate we have 96 organ chip can be used to test the drug. And with this organ chip, we can culture cells in organ chip, and then this is 2-channel organ chip. We can culture cells in one channel with another channel to deliver the nutrition or drug treatment. After give treatment for a few days, and then we can collect the data to a different type of data to evaluate the treatment effect. And like the images, like bright field image or fluorescent image, high content images. And this you can see this is the fault because the 3D model we have 3D images. And also can collect the biochemical assay data and the omics data to monitor treatment effect. And with this data, we can build an AI model to understand the treatment effect. Actually, we can, for example, for the imaging data, we build an AI model to understand, given the drug with a specific mechanism of action, like we know the drug's target disease, what target interact with, what's the pathway, what disease to treat, and what's like the target on which anatomy. And then with our information, we'll see how this drug causes the change on the cells. And then also based on with new drug, based on how the cells react to the treatment, and we can get information like what type of drug, what type of drug works, like is what type of target react, and it may be used to treat what kind of disease, what pathway is involved. So at Zelo, we develop things in different steps. In the first step, we just use to do the wet lab experiment to test the drug efficacy and safety. And we use organ chip to test the efficacy safety. And then we link the result, the organ chip result to the clinical outcome. And after we collect more data, we can build the in silico model, which is directly predict the organ chip result in the computer. And after that, in this case, you don't need to synthesize the compound. When you design new molecule, you do not need to synthesize the compound. You can directly make the prediction and then choose the top 10 and top 5 synthesize it and then test this in wet lab. And the wet lab result can help us verify the prediction and also improve the model, AI model. And after that, this AI model may be used to optimize the drug structures to make a better drug. So before the end of this talk, I also want to mention about this. If we want to develop this AI solution in drug discovery and development, probably not only develop solution in drug development, we need to first make sure we can define the right problem. And this is where, and also we collect the right data and develop the right method. The three components actually need to be do together. Is you cannot separate one from other one. For example, if you want to define the right problem, and then first, you need to know what kind of problem can be answered by the data and available method. And also for this problem, what's the context of use and what's the impact solving this problem? And what kind of performance we need to achieve. More importantly, we need to convert the problem into the problem for the AI modeling, like what's the input, what's the output, what the metrics we should use to optimize the model. I think many times people make a mistake on this part, that they cannot clearly define the input, output, the metrics, and this causes a lot of issues when we develop the AI solution. And this one need to have very good communication with the domain expert. For AI scientists, you need to have very good communication with the domain expert to define the problem. If you can define the right problem, probably you solve the half of the problem. And second thing is to collect the right data. And this also, to collect the right data, we also need to know what problem we want to solve and then what method available to solve the problem. Based on that, we collect the data. And also need to understand the data because there's so many different type of data. What's it really measure? And what's the strength and the weakness? And how the data was produced and collected? What's the potential issues and limitation of the data? And also, I see lots of publications, and even the very high-end journal, and they use the wrong data to build the model. And third one is develop the right method. Actually not the complete model always is better. Not always the AI model is better. If we can solve the problem with a simple method, we always need to use a simple method. Like if a problem can be solved, use the statistical analysis, and probably no need to go to the neural network. So yeah, I think it's the talk I have today. Now I'm happy to answer questions.
Oh, thank you very much, Dr. Xie. This is a very comprehensive to address the drug development process and combination, how to use the different tool and different methodology and imaging, all these things. Put all together is a tremendous work. So appreciate it very much. Thank you. So now I think that Dr. Xie gave that very important talk, and that also attracted a lot of audience to have. And we have a lot of questions already. So then we start to have this Q&A session. And let
Dr.
me,
Yang, sorry
yeah.
to interrupt, but would you like to open your camera? You don't have to if you don't
Oh,
intend to.
that's okay. Yeah. I think the to address that uh, uh, audience question first. The first question is ask you how to use the AI to choose the right patient.
Oh, yeah, that's a very good question. And actually, a few things. First, for the recruitment, and like previously, we always based on some testing result and measurement to choose the patient. And right now, also people explore the use large language model to based on the clinical notes to choose the patient. That's the first thing. And second thing, also we need to probably can use AI to identify the biomarkers to choose a patient. Actually, two type of patient. One is uh, responder, non-responder. We all
Hmm.
want to find the patient for response to treatment. And also we want to find the patient have less safety issues. And this also, always based on the different type of data, like the omics data and other data like the imaging data, and you know to identify what type of patient response to treatment, or what type of patient have the safety issue. That's just for this one type. Another type we call the sometimes we call the enrichment study. The idea is sometimes for the chronic disease, it take long time to see that because the disease
Hmm.
progress very slow, and then it take a long time to see the treatment effect. And in that case, can we use AI to find the patient and based on the imaging data, based on omics data, and based on other measurement to see like what kind of patient that progress faster than other type of patient. And then if we enroll this patient in the clinical trial, it's possible to see the treatment effect in a relatively shorter time than a long time. If you can cut your clinical trial from 3 years to 2 years, you save a lot.
Yeah. So usually when you do the clinical trial, in your protocol, you have the inclusion and the exclusion point. So you did from the clinical trial design, you have that, what kind of a patient you are looking for. So from the inclusion and exclusion, then how they can combine with the AI tool. You use these inclusion and exclusion criteria to set up the AI to kind of machine learning to select the patient.
Yeah, yeah, I think it's also, it's a very, it's a good question. Based on my previous answer, first thing is, for exclusion area, probably first thing we use AI to define some biomarkers, that
Hmm.
if you want to find us for different purpose. One is if you want to find some like a responder, non-responder, that kind of things, we need to first use AI tools to find the biomarkers, then use that to define the inclusion and exclusion criteria. That's one thing. But most time when we talk about the enrollment, and
Right.
so in that case, we already have actually there's also AI tool can another thing AI tool can help the define the inclusion and exclusion criteria is two ways. One, I have worked on some project. One is uh, we can use AI to find out previous studies
Hmm.
what kind of inclusion exclusion criteria was used for this disease and this type of treatment.
Hmm.
And second thing is also based on this inclusion and exclusion criteria, and also from the real world data, we can find out if we change a little bit of how to optimize this inclusion exclusion criteria. Like sometimes if you change a little bit, and then you can increase like a population. Uh, a lot. Like you can increase your speed of recruitment, and then you can think about it, should I modify my inclusion or exclusion criteria? And third applications, if we already have the inclusion exclusion criteria, and many times, to find the right patient is not straightforward. Normally people directly compare numbers to the inclusion exclusion criteria, but most time in the real world data, and they may not have this accurate numbers. And then use AI, you can go to actually many people work on this field. They use natural language, large language model, and to goes to the clinical notes and to
Mhm.
find the patient match
Mhm.
based on clinical notes to find the patient match your inclusion, exclusion criteria, and use that to select the patient.
Okay, thank you.
Mm.
So next question, yeah, from the audience that they ask, what metrics do you use to evaluate the effectiveness and accuracy of AI integrated into workflow? Can you provide an examples?
Yeah, I think it always depend on the problem or the application, the context of use. Depend on like what kind of application you are use this for. And for example, I have worked on a project to screen and probably it's for toxicity studies. In animal studies, we collect the data to see if like the pathology image and to evaluate if the drug caused any damage on the tissue. But most time there's no damage, it's normal data. And then if we want to screen this, and if we have AI to screen this, and like only like a pathologist review the images or tissue which is suspected to have an issue, and we can save a lot of time. But for this type of work, we need a high sensitivity, because we don't want to miss any positive case. But specificity and even lower specificity is okay. Maybe like for example, we need probably 95% of sensitivity or maybe 100% sensitivity. Specificity, maybe 60%, even 60%, we can still save 60% of time. So they all depend on sometimes probably we need a high specificity, lower sensitivity. And also same thing for the accuracy and for other performance we need to carefully define what we want to want to measure to evaluate the performance.
Mm-hmm. Mm-hmm. Yeah. Mhm. Okay. Thank you. The third question here is how to identify and fill the data gap across the data set from the different metrics.
Can you repeat the question?
How to identify and fill the data gaps across the data set from the different metrics.
Uh, yeah, I think it's probably, few things. One is, right now previous is, I'm not sure if it's from different metrics means standardization or, you know, harmonization. The,
I don't know what
yeah, I'm
that
not sure the question,
different
but
metrics
anyway,
means.
yeah, so I think that for data,
So, Alan, could you just open your the phone and microphone and just directly talk about it? Alan, are you there? Okay.
but anyway, I will answer some of the questions to Cooper, So I think it's also interesting question. So probably the first data gap, like, we have data, first thing, we did have data collected from historical, from different type of device, different
Hmm.
technologies. And this makes
Hmm.
it very hard to directly use them together to train the AI model. And, once few things, one is, if possible, we can do the standardization, harmonization of data. And secondly, we may train the models based on each individual data set. And then we can think about how,
Hmm.
we get a final measurement and then we can combine that together. And also right now that
Hmm.
some technology, we actually one thing I have to see is previously many data we think is not very useful, and now with the technology we find it's useful. Like for example, just I mentioned like all the text information without any labels. Right now with the self-supervised learning, we find this information also is useful for us to train the AI model. So another thing, if the data come from different modalities, also the technology probably can help us to link the data together. For example, the knowledge graph. And you can link the data through a knowledge graph. You can link the data from like the different modality, like the omics data and imaging data, or maybe the data for target, data for for phenotype, and link them together.
Yeah, yeah. I think that actually, I feel that question is very important because sometimes, as you mentioned that before, when you get the data from the animal data, and that could be has a difference from the cell lines data, and that also can be different from your clinical data. So that could be the big gap between the data set.
Yeah.
And at that time, and how do you interpret and how do you feel that gap?
Yeah,
That's
actually, that's what we normally want to do is like we use the in vitro data.
mhm,
We use the in vitro data to predict the animal study result and the clinical result. That's effort in the build this, and also from animal result, how can we predict? You use animal result to predict the clinical result outcome. That's the goal. If we can do, if we can get that, and then before we get into animal study, like in vitro study, we always have more confidence about how this will works in a later stage. So that
mhm,
can say
mhm, mhm.
increase the success rate of the clinical trials.
Okay. All right. So move to next question. Could you give uh, could you give us example of your last page? So this is your last page. Now she fully understood what's the example for the last. Talk about the how to integrate all these three area problem data methods. Do you have such a examples?
So yeah, I think for example, few example for one example is for the matrix, what for to define the right problems, we need to define the right matrix. Just one example, many people right now, also the challenging program, also many publications, they try to use the like HE image to predict the single cell gene expression. And but for this task, right now many people use try just directly measure the difference about between the predicted gene expression and the real gene expression. The problem is we know if in biology we know some genes high expressed gene and low expressed gene. If you directly use for example, if you use the square difference, and then the high expressed gene probably will dominate and your matrix. And another thing we also know some genes the co-expressed, they increase or decrease together. So if you use this matrix, probably the group of genes, they have a large number of genes that co-express together, that will affect when you more optimize your model based on that, probably you always predict that type of gene more accurately than other type of gene. But if we want to do the biomarker development, probably most time you're not working on that type of gene. A larger group co-express together. So, and also collect the right data part. And like another one example is, a few years ago, also the very, the publication for from very famous university that they have AI model identified the antibiotic bacteria drug. And for the drug safety, they use the data, which they use the data failed in clinical trials because of safety issue. So they give a label like a positive or negative. If the drug failed in clinical trial due to safety issue, that's positive. And if the drug passed the clinical trial, it's negative. They use this data to train the model. But the problem is they're not aware that the drug failed clinical trial due to safety issue always also have link with the indication, like which disease you are treating. If the drug, same drug have same type of safety issue, if you are treating cancer, probably they're okay with this safety issue. That you can pass. But if you use to treat to reduce the body weight, probably will not pass. So if you don't understand the data, and then probably use the wrong data to train the models. And also develop the right method part. We saw a lot of stories, and people just use they have a hammer, and then they just try to find the news with the hammer, and they always use a very complex models for some very simple problem. And actually, my experience, I saw a lot of times, many times in drug discovery, we have deep data instead of bigger data. I mean, the deep data, I mean, we have a very small sample size, but each sample we measure many things. Like gene expression, we may have 20,000 gene expressions, and
Yeah.
also have protein other data, have very high dimension data, but
Yeah.
small sample size. And in this case, probably you want to find some method, is robust and how to combine them together, like different, like omics, if you have gene expression, if you have protein or different data, how we can combine them together, integrate them together. And also find a lot of times, sometimes even like a linear regression works better than like a machine learning or deep learning method. So we have to based on the problem, based on the data, and to choose the method.
Yeah. Okay. So that's to answer that question, to integrate the problem data methods. So that's one. So next couple of questions is focused on the organ chip. Okay. So uh, the the audience ask, hi, I'm wondering whether current AI analysis based on the organ chip still suffers from the systematic error. How are we convinced that an organ chip is a good representation of an organ? With different results in individual organ chip. For example, biological re- replicates. Would we be more interested in precise stratify patient, hence precising medicine, or further generalization? So that's the question. One is actually it's a two question. One
Oh,
is
yeah.
about how can you make sure that the organ chip you use is really a true representation of the
I got the
organ.
question. Yeah, I can answer the question. That's
Okay.
that's a very good question. So, first, for how how organs can really represent the real biology in real systems. Actually, we have to aware uh, organ chip is not a real organ, and it's not normally when we do this, we build a disease model. It's actually all based on the specific context of use. Like, do we want to use for this like to test the liver injury, or do you want to use to test the cancer treatment effect, or even the cancer treatment effect? Do we want to use for chemotherapy, or want to use for the like the immunotherapy? For different purpose, we need to develop a different disease model, and then we use this model to test the drug. And normally also when we do this, we always, after we build the disease model, we always use the existing drug to test that. We know which drug works, which drug doesn't work, or we know which drug causes liver injury, which drug does not cause liver injury. We use that to verify the disease model. And also the AI we use here is we use AI first to quantify the treatment effect, like how cell react to the treatment. Also, we use AI to characterize the effect. And for example, we can use AI to learn the representation from the image of cells, and then we can build the link between. Based on that, we can build the link if cells change it in this way, and that always mean it may cause like a liver injury. If cells change or maybe cells change this way, always means we can suppress the cells, we can kill the cells. So that's how the AI would use that. And second question about the precision medicine. And we also did a lot of work on this. You can get like the cancer cells, not only cancer cells, you can get biopsy from patient. And we have some collaboration in China, get biopsy from patient and then use the cells from the tissue and culture cells and tissue in the organ chip, and then we test which drug can suppress or kill the cancer cells more efficiently, and use that to help, and after we get this data, we compare this result with the clinical outcome and to evaluate if this model works to help the physicians to select doctors to select the right treatment.
Okay, thank you. So next question is also related to the organ chip. We used to do the drug repurposing using the cellular or organ chip phenotype assay data by informatics. How is the AI different from the conventional bioinformatics? So you
Yeah.
use the organ chip to get the by informatics to get the phenotype assay data. How that different from the conventional?
So yeah, I think from AMS part, no big difference. We always, for drug repurposing, you can use real world data, find some clues. You can always use omics type of data or use knowledge graph to find out like which drug may be used. For example, we can use a knowledge graph and use AI on the knowledge graph to find out if this, actually knowledge graph can do the link prediction. So, many times the knowledge graph will not give us all the information, all the knowledge we have. Because many times, like if the drug never used to treat that disease, we don't know whether or not this drug treated the disease. So knowledge, with AI technology, we can do the link prediction to discover and to identify and discover the link between the nodes, like between drug and disease or between drug and target. And after we have that, the organ chip may be used to test this. First before we test this in animals or in clinical study, we can first use maybe organ chip to test this. And organ chip give us additional data. Organ chip just give us data is a way to provide the data and to verify something. So we can apply AI technology on this data and to verify if this drug really treated this type of disease.
Okay, thank you. Next two questions still focus on the organ chip. So lots of
Yeah,
people
that's good. That's good. A lot of people have
interested in the
Sorry,
organ
Dr.
chip.
Yang. Dr. Yang, I think Dr. Wu Chao has raised his hand for quite a while. I'm not
Oh,
sure if you noticed that
I
or
didn't
not.
see that. Okay. He wants to talk?
Okay, thank you, and thank you, Dr. Xie, and
Yeah, please.
Okay, so this is Chao from Maryland. I really appreciate this talk. Thank you very much. And for myself, I'm a data scientist working on the agentic AI in the surface area. And I think you are working on the research and the drug area. So one question for you, how do you envision the regulatory, like FDA, will they change the approval process based on the new approach for drug discovery?
So I think first thing is regulatory like FDA. They adopt the AI technology very quickly. Actually, probably 10 years ago, when I have a meeting with FDA and to discuss some technologies, that time they always say we don't like the black box. They show some attitude is not, AI is not something that we adopted, in the near futures. And right now we can see the start, that they also did a lot of research on AI. They start to use AI in their work. And so, but still, I think that for the AI, not only for AI, also the, regulatory FDA also recently they also announced some policies about organ chip use and also other technologies for to replace animal studies. But anyway, this it's not happening overnight. I think most time we always work with the regulatory. It's also depend on issue, case by case. Normally, if it's new, if it's emerging technologies, we work with the regulatory together and to define the problem and how we approach that and how can we standardize the process and then make it, in the real process of into the real process of drug discovery and development. So I'm not sure if I answered your question.
Yeah, thank you. I think you answered my question. Thank you.
Thank you.
Thank you very much, Chao. I very welcome you actively participate in our the discussion and the AI series. So let's continue the question. Do you have any more questions, Dr. Wu?
No, thank you.
Oh, okay. So let me continue to address the question that the audience came from the audience. In practice, are there widely accepted or open source in silico model for predicting organ chip assay results, or does each lab typically need to develop and validate its own AI model? If so, does someone within the lab itself need strong machine learning or computational background, or is it common to collaborate externally for this part of research?
So I'm not very sure to understand all the questions, but one thing that for the last questions, the answer is definitely it's good to if you have an internal expert, it's always good to have a collaboration with internal expert. Make sure, like many times we have the like open source tools, and also maybe either open source or commercial tools. But I think it's always good to have, if you have the internal expertise and work with them together and you can learn from each other. And first thing, you can make sure you use the tool correctly. And also you can learn from each other. My experience always, the AI experts always want to work with the domain expert to learn the domain knowledge. So the domain expert also is good to work with the AI expert to learn more about probably not the exactly AI technology, but what the AI can do for them and what's the correct way to use this type of tools. So that in this way you can learn each other, and then probably in the future you will also become an expert in this field. So we also said like previously, in the imaging radiology field, we also see AI is not replace radiologists, but the radiology, the knowing AI will replace the radiology not knowing AI. So, but anyway, I think if you for the people in the lab, and then also I think it's good to learn how to use these tools because it's coming into our life, especially over the last few years. The AI tools come into a personal life very quickly. And previously probably not many people use AI. Right now, large language model many people use this every day. They did improve their work efficiency.
Okay. So another question is still about the chip. So the question is, chips and organoids could reflect some, but not all human in vivo feature. Is there a successful case to use the verified feature while the neglecting other non-reflective ones? If no such case, is this ideal feasible?
Yeah, I think for now,
No.
I will say, as I mentioned earlier, for any applications, we need, either the organ chip or AI diagnosis tools or any tools. The key thing is the context of the use. So normally we always need like like AI for medical device or something. We always clearly define what context we will use the technology. And this, normally it's not like this context is a narrow context. So like if we want to use, if when we use the like organ chip and then can clearly define, we only use this organ chip on this type of disease, and even this subtype of disease, and with this type of treatment to test this type of treatment. So in this case, and we can test if it works or not, and use that, use the evidence to show the community, to show the regulatory, and if we can show this works, and then we can use that. But we never think about like use the organ chip and for the general purpose to use.
Yeah, yeah. So it's a very specific case. Actually, this question is also I have the same feeling about that because in principle, in vitro and in vivo, that's a totally different case. All the biological environment and the material is totally different. So but got to be very careful about how to use the organ chip. But fortunately, I think the today is the third talk about the AI the applications. But if we move to the fifth talk, then we will invite the FDA's reviewer to talk about their policy from their policy viewpoint to how to evaluate the organ chip. So just stay tuned, and we will hear more discussion with FDA's officers. So let's continue our discussion. The last question here is the one person he previously asked about the bioinformatics issue. And he say, of on my previously repurposed AI more than traditional bioinformatics analysis when it comes to the drug repurposing using either cell-based or organ-based chip for the phenotypic screening.
So you break
He just, to emphasize what is the AI to add more value than the traditional bioinformatics analysis.
So yeah, and because you break out lots, so I'm not sure I understand the complete question. But for the AI and bioinformatics, definitely, if for a small problem or question, Many times we still use, as I mentioned, if you can use a simple method to answer the question, we do not go to the complex method. And for the bioinformatics, the very powerful tool, many times we can directly use that to answer questions. But now I think that with the AI agent, and previously what we do is we have a specific question, you need to ask a specific question, then you can find the bioinformaticians based on the questions, write some code, and get the answer, do the analysis, get the answer, and then give some plot like a visual, or, maybe PowerPoint slide, Word files, give a report. And right now with AI, and actually make the informatician's job much easier. As I mentioned earlier, biometric AI agent from Stanford Group, they have they incorporate all the bioinformatics tools in the system. And then when you have a question, these tools can automatically, first you can tell which data set you want the agent to do the analysis. In the meantime, this agent can also maybe goes to some database, existing database, and after analyzing your data, they can compare this result with the existing data. And then, maybe call different AI agent with, also from publications, from, other resource, they can put all the results together, give you more comprehensive, answers for your questions. Make it possible. And then as a bioinformatician, you much easier, you can probably spend more time about first verify if the result is correct. Secondly, how you want to interpret the result and how you link the result with real life problem. You can pay more attention to the bigger pictures than previously, like spend more time writing the code. So I think it's probably better tools for the bioinformaticians.
Okay. All right. So that's one. I also have a question regarding the organ chip. Do you know is there any currently is there any commercial organ chip available, and how they to define the organ or define the disease or define what's the purpose of these organ chip? Is there any commercial available
Yeah, I
these?
think right now a lot of company
Uh-huh.
doing organ chip that offer the service, either sell the organ chip or sell the service. Some company that directly sell the organ chip device, and you can use the device to build the disease model yourself. And some company that provide the service, like you can give a drug to them, they test the drug for you. So yeah, I think right now the many options available.
Okay, thank you. So continue the question. If you could recommend a clinical AI agent for the drug discovery in cancer cell therapy, and why?
So
Could you recommend a clinical AI agent for the drug discovery in
mhm.
cancer and cell therapy?
So I think,
Why?
first, there's some like already have some AI agent available, like the one I mentioned in this talk from Stanford Group. But I still always think, if you have the resource, and then I think it's good to, like the biotech or pharma, it's good to build the agent because build the agent is not right now is not that difficult. So build the agent based on your scenario, based on your need. And
Mm-hmm.
so, that will be, I think this, always not, probably sometimes not, for some tools, you can direct get off-shelf tools, like commercial available tools to use. And some tools is good to adopt especially if not take too much resource, and then you can, put the things together based on your, uh, with this, put your use scenario, your application in the center, and then configure the AI agent for that purpose. Otherwise, we have when we build the product, or we have open source, either open source or commercial, and probably the product is not developed for your purpose, and then it's not centered on your applications. In that case, you'll probably always find out, I love to have this function, or I love to have that features, but they don't have that. So, if that, develop that product is very expensive, costs a lot, and probably you can have a collaboration with other, external partner to develop that. But if cost is not that high, and especially if you need the flexibility, you can always, build the agent yourself.
Okay. So either most of that is you based on you build on your own, or you can to use some available agent. You need a search. So the person asked another two questions. One is regarding the explanation of the learned uh, latent space for new biological mechanism discovery. How developed is the field now in this regard?
So I'm not sure it's
Yeah, I didn't really.
the explainable or interpretable AI. And so I think as the different technologies developed to try to make this, I think there's also a lot of framework available try to make the AI system interpretable or explainable. And like if you are with the, these models and like after prediction also give back the results like this come from based on what kind of measurement or what kind of features. And if you change the features, how the change will affect your final prediction or measurement. So there's also different technology on this part. That's, still is, I think everything have to be used back, based on your application scenario. And sometimes, we only need the result, like if we can see the result and we can make a judgment. Like, for example, image segmentation. After we see that, we can clearly see if the segment is correct or not. In that case, and we probably don't need to worry too much how they make the segmentation. But if you for the patient selection and diagnosis, if you cannot tell why you think this patient was definitely have this disease and why you select patient for this treatment. If you cannot tell them the reason, and then probably they will not listen to you. So in that case, it's good to have the interpretable AI to trace back, the reasoning status, like the reasoning chain, and then back to the original data to indicate this decision is based on what a pattern of the data. So then the expert, the doctor or physician, they understand is this reasonable or not reasonable.
Okay. The second question is the data availability for the in silico drug test. Is the current data enough? What are the key data that's lacking? What would the ideal data look like? And that's to a
Okay,
second question.
sure, yeah.
The current data, is it good enough?
Definitely the data is not enough. It's way not enough. That's why I mentioned like, many organizations, because ideally we want a lot of clinical data. Like
Mhm,
all the chemical structures, if we have the clinical data, show the
mhm,
efficacy, the safety, and
mhm,
then we can directly predict the clinical outcome in silico. And
mhm,
that would be very, very useful. But that's impossible. We cannot test, the chemical compound. We only chemical compound if you want to test on the human, you have to pass the like in vitro study, animal study, show the safety, and then you can test on the human. So
mhm,
that's why we use organ chip. This one also people use the in vitro study, like high throughput study to
mhm,
systematically collect the data to build a
mhm,
model. And we use organ chip to collect data to build the model. Although it's still not a clinical data, and we hope this can link, can have better prediction or have better predictive value, clinical outcome.
Okay, great. Uh, I think that's all the questions from the audience. And if that's the normal question from the audience, then the Q&A session just stop here. Let's give applause to Dr. Shea. I think it's an excellent talk, and also to address lots of questions from the audience already. Uh, and thank you, Dr. Shea.
Yeah, thank you. Thank you for the invitation, and also
And
the audience
yeah,
for attending this seminar.
yeah. So could you get out from
Yeah, I'll stop share.
yeah, screen share? So let's back. So today we have the Dr. Xie give a very nice talk. So we also have further, could you share, could you see my screen?
Yeah, I can see it.
Okay. So next Saturday, we have another important activities, and we will have, as I mentioned that eight professional organization in DMV area will organize AI innovation and entrepreneurship workshop. And we invited Dr. Tang to give the agent agentic AI the history about that agentic AI. So in the future how that will develop it. And we also invited the CEO of Tedco company, which is Maryland, the major Maryland investor firm, CEO, to talk about, in his view, about the innovation and the advanced technology in Maryland area. Then we also have Kevin Hall to give his leadership pitch, and afterwards we also have the expert to have the comments and to comment on his leadership and how to that we hope that through this activity and through that one is really can help the young the entrepreneurship and to start to have the more even innovation and entrepreneur in Maryland area, in Maryland, Virginia area, as well as the whole nation's area. So I think this is a good to encourage everyone to participate. So this is a give a pre announcement, and I think the Dr. Yang, another Dr. Yang, will be the host for that. So, Dr. Yang, do you have any other to add to?
No, thank you for that announcement. I really welcome anyone who is interested in AI, especially agentic AI, which I think is the very much hot topic today and even in AI industry. And this is also very much about startup entrepreneurship and investment. So yeah, I think it'll be a great event, and welcome everyone to join us.
Okay, so that's next Saturday, and that is we not only on the line, and also we have the off the line insight. You can if you are in the DMV area, you can go the place to join us. And so afterwards, we also have the lunch and the social activities so we can know each other, discuss the technical issue, and also make more friends in this business. And the other one is next month, uh, the August. Then the August we invited the Bo Song, Dr. Bo Song, and he is also a very important person, and we are very lucky to invite him. He is the MD and the PhD, and also he is further. His talk is well focused on the diagnostics and specifically in pathology diagnostics. So that is also related to this digitization and AI, all these big issue, all connected to these, the diagnostic and the pathology study. So this is in August. So I hope that everyone can enjoy us further, that we can learn more about the AI in the drug development and in biopharmaceutical industry. So with all these announcements, and I like to thank you for everyone, and to especially thank the Dr. Xie for his tremendous, very important the talk, and give us the very deep vision about how to AI involved in the drug development process. This is one, and also we appreciate so many audience and to join us tonight, and so far still have more than 60% to with us. So I hope everyone can learn something and to improve in your work. And also, I hope that everyone can join us for the next activity and next month's activity. So this series talk will continue our efforts. So thank you, everyone. Good night.
Thank you. Bye.
Thank you. So we close here. So let's wait a couple minutes.
呀，我们自己人要留下来
你等一等，
聊聊吗？
你等一等。